Good morning, everyone, and thank you for joining us today. The team at Teledyne Photon Machines is absolutely thrilled to continue our recent webinar series, which has explored a range of exciting and fast-growing applications across our community. The response we've received has been phenomenal, and we're genuinely energized by all of the new interest and engagement that these sessions have sparked. We're excited to keep the momentum going well into the future. Speaking of exciting applications, I can't wait to share with you more about today's incredible topic of dendrochronology, a field that blends precision analytical instrumentation with the remarkable stories recorded in trees themselves. It's a fascinating intersection of science, technology, and environmental history, and we're delighted that you're here to explore it with us. Before we dive in, I'd like to introduce today's speaker, Soumaya Belmecheri, and my colleague, Garry Armstrong from CIRCON. Both of them, thanks to both of them for taking the time to share their expertise. Garry has kindly joined us as an expert in the technique of LA-IRMS and will be helping with the Q&A session to follow. He is himself a seasoned mass spec professional with more than 25 years of experience across engineering and commercial leadership roles in analytical instrumentation. Starting his mass spec career in 2002 at Micromass, he developed a deep expertise in both organic and inorganic mass spec. Today, as a director at CIRCON Instruments, he plays an important role in advancing high-precision isotope ratio mass spectrometry used by leading research and industrial laboratories worldwide. Our featured speaker today is Dr. Soumaya Belmecheri of the University of Arizona. Soumaya is an associate professor in the Laboratory of Tree Ring Research, where she studies long-term environmental variability during the Common Era as recorded in terrestrial ecosystems. Soumaya's research explores how these environmental changes shape atmosphere-biosphere interactions, with a particular focus on seasonal hydroclimate variability and extremes, including hydroclimate whiplashes and tropical cyclones. We're delighted to have Soumaya with us today to share insights from this important and truly compelling area of study. Before we get started, just a quick reminder about questions. You're encouraged to submit your questions throughout the session using the Q&A session at the top of the screen. We'll gather everything as we go and do our best to address as many questions as possible at the end of the presentation, time permitting. Of course, if we don't get to all of them live, we'll make sure that every question receives a follow-up response after the webinar. With that, I'd like to thank you all again for being here, and I'm happy to get started. Soumaya?
All right. Hello, everyone. Greetings wherever you are, which time zone you are in. Thank you, Lucas and Garry, for organizing this and for giving me the opportunity to share recent research and actually my excitement about the laser ablation coupled with an IRMS system that we have installed recently. I'm going to share my screen. And all right, let's do this. Okay, and up. Can I have a confirmation that you can see everything, or you can see everything well? Good. All right.
Good.
Yep. Thank you. So the title of my talk today is At the Scale of a Ring, Unlocking High-Resolution Climate Archives in Trees Through the Laser Ablation IRMS System. So what I'm going to do today is give a general overview of how we thought in the past and how we are thinking about tree rings as recorder of climate and environmental variability, and highlight two recent applications using the newly installed laser ablation system in the Laboratory of Tree Ring Research. All right. Well, let's start from the beginning, right? I'm showing you here a cross-section of a tree ring. The pith is over here, the bark is over here, and these concentric features represent annual growth rings, which basically they correspond to 1 year worth of growth, right? So you can also see that the succession of these concentric tree rings have different or variable width. Some are narrow, some are wide, and what they do reflect is the variability in the most limiting factor of growth of a tree. For instance, if you're in Scandinavia, that will be temperature. If you are in the Mediterranean area, that will be rainfall. So depending on their location, tree rings can record a variety of environmental signals. And for a given chronology, let's say whether it's ring width, which is the most common proxy or indicator we use in dendrochronology, or if we measure the relative abundance of carbon and oxygen, the most common stable isotope measures in tree rings, you can relate the variability of isotopes or of the ring width to rainfall or to temperature. You can use either a statistical model or a process-based model, and the goal is always to reconstruct back in time the variability of rainfall or temperature or any other environmental factor that is recorded in the growth rings or the isotope values within an individual year. So for a long time, our unit of time in dendrochronology is 1 year. If we're thinking about one ring, it corresponds to 1 year worth of growth, and which which results in an annual resolution. Most of the studies of past climate variability or environmental change were based on an annual resolution. However, from the image I've shown you before, you can see that there was an alternation of sometimes darker and light colors that represent growth that occurs in the spring or in the summer. And these correspond to two different parts of the ring. The early wood, which is characterized, is shown here in this picture, which represents wood anatomy. The early wood, which represents the growth that occurs early in the spring, often for conifers, and is characterized by large cells with thin walls. And the late wood, which corresponds to summer growth and corresponds to smaller cells, however, thicker cell walls. That's why the darker color of the ring that we observed in that cross section. And we have been applying stable isotope measurements on early wood and late wood, but also ring width, measuring the late wood and early wood, which gives us insight into seasonal variation. So we moved from annual resolution to seasonal resolution. However, recent advances in a variety of techniques, including quantitative wood anatomy, is moving us beyond a seasonal resolution, but into a sub-seasonal resolution. We are down to the cell. So this is an image that shows further the wood anatomy, and you can see the early wood and the late wood from a ponderosa pine from southwestern United States. And we are down now to the cell where we are able to measure anatomical features such as the size of the cells, but also the thickness of the wall, and link those to variation in environmental condition. So it's given us more granularity, a sub-seasonal resolution, and opens an opportunity, if we have technological advances, to now look at short or extreme variation in the environment, such as tropical cyclones or frost events. So we are now well positioned with technological advances, one of which is the LA-IRMS I'm going to be talking about later, but also using quantitative wood anatomy, is to now study climate variability, environmental change from the cell to the global scale. We are able to link cell level processes to global level processes such as a jet stream, for instance. But also it allows us to link subseasonal resolution to multi-decadal. So we have a level of temporal depth and resolution that has been unprecedented in the past, combined with this amazing technology such as quantitative wood anatomy and the capabilities to measure stable isotope variations at a finer resolution than the seasonal one, which I'm going to detail in this talk. All right, so let's talk about the laser ablation system that is installed in the Laboratory of Tree Ring Research. I'm showing you here a picture, me pausing and very happy with the system that we installed in January of 2025. We have a laser ablation coupled with an IRMS, but we also have an EA, a pyro-earth, and this is part of a new facility in the Laboratory of Tree Ring Research, which is called TIME, and TIME stands for Tree Ring Innovations in Mass Spectrometry for the Earth System. The lab now is fully operational. We have capabilities of to measure carbon and oxygen through the Pyro-Earth EA, but also when it was installed, we had capabilities to measure delta 13C using the laser ablation system, which is coupled also with the CryoFlex system shown here in the picture. However, a great update and progress is the installation of the method to also measure simultaneously the oxygen 18. You can see me here in a picture with my colleague Elina Zalstedt, who pioneered this method and which was published in, I believe, in the spring of 2025. We successfully installed this in the Laboratory of Tree Ring Research in December of 2025, and we are now able to simultaneously measure carbon and oxygen using the laser ablation system. And I'm going to provide two examples of this recent application, and then we will have time for further discussion and questions from the audience. All right. Just a quick overview. To my knowledge, laser ablation IRMS systems that are fully focused for tree ring applications, and I ordered them here, I think, by chronology, and Garry, please correct me if I'm wrong. There's a system that has been installed and fully operational, successfully operational in Luke, Finland, Swansea University. So in Luke, Finland is with Elina Zalstad, that you saw in the picture, who assisted with the installation of the oxygen method. Swansea University with our colleague Neil Lauder, WSL in Switzerland with Matthias Saurer, the University of Arizona, the Laboratory of Tree Ring Research, and recently the University of Bologna in Italy with my colleague Rosella Guerrieri. All right, I'm going to just give a brief disclaimer before I move forward. Most of the data I'm showing are in publish, is work in progress, is part of ongoing collaborations. So please do not record or photograph, screenshot, distribute, or share any portion of the results and discussion without prior permission. Open the floor for questions. I'm happy to answer questions about these two applications, but also more technical aspect of what was involved in the analysis. I'm going to stop sharing my screen. Thank you.
So thank you, Soumaya. That was absolutely wonderful. And as a simple-minded biochemist, I think remarkably humbling. Lets me know just how little I know about your application, but I do find it incredibly interesting. So thank you for that. We do have some questions. The first one is probably a really good one for Garry, as it involves the technique. The question is very simply, what does laser ablation produce? Is it CO, CO2, and what about fractionation?
Okay, so in the early days when this collaboration was formed, the system was set up primarily with carbon measurements in mind, and the technique was to ablate some material and then combust to CO2. So for pure carbon measurements, the preferred technique is to do a combustion to CO2. In the latter technique, where we're looking at oxygen isotopes, there's been a lot of work developed by Alina's team and now Soumaya on producing CO in situ. So we don't need to have that combustion or pyrolysis stage in there, which allows things to be a little bit cleaner, produce some good, nice data. And one thing we found with both techniques is there's a lot of latitude for the researcher to play with this because of the way we are evacuating the sample chamber, sweeping out the material of interest, and then passing it through the interface before it hits the IRMS. You have tremendous variability in laser energy, spot size, ablation time, and yet we still see no fractionation, no perceivable fractionation in the measurements. So it's a very robust technique for both oxygen and carbon measurements.
Yeah. If I may add to that, and thanks for the question, actually, yeah, I mean, because we're measuring wood, right? And the CO is produced, there is a little bit of CO2, but the CryoFlex interface is amazing because it allows us to control for that, to trap CO2 and the chromatography that comes after to separate nicely the peaks. And we've done a lot of tests with the variety of standards from C4 and C3 plants, and we can see that, it's very reliable, it's very consistent, and no indication of fractionation. And we also tested material that was measured in different labs.
Excellent. The next question is, what is the analytical precision or uncertainty for the the oxygen isotope, and what standards do you utilize?
Okay, great question. So actually, I am surprised how amazing the analytical precision is. And I'm saying this, for the oxygen being a fussy, I will say, isotope to measure. So far, on the laser ablation system, measuring oxygen 18 on the CO produced from the ablation, we are anywhere between 0.1 to 0.2 per mil. And similarly for the carbon, actually, it's even better for the delta 13C. For the standards, this remains I would say like a work in progress, and there's the list of 5 locations where we have similar system. We have been having conversations about it, but we are using the traditional IAEA C3, right? It's cellulose, so we're using cellulose standard for wood measurements. That is not available anymore to purchase, so sheets, right? But we've been also developing our own standards. For example, in a lot of these runs, I used corn husk. So especially to give us a possibility to do a 2-point calibration in the system. We've been also using the VWR cellulose pads. So the idea is always to characterize them in a traditional EA system before assigning them values for in the laser ablation system. But that said, there are wood standards. For example, the USGS standard is available. We use it traditionally in a classic EA setup. In the laser system, it tends to be a little bit more tricky because that wood is darker and might cause problems in the sample chamber. So we're sticking so far with alpha cellulose pads and also corn husk or yucca for to have a 2-point calibration for the carbon. You have to pelletize them yourself, so we need to do that. That's part also of I would say, not the limitation, but it requires the equipment to do so.
Thank you. I think we've got time for one more question. We've got one regarding spatial resolution. It says, when conducting LA-IRMS analysis of tree rings, the width of the rings vary from year to year. Can you accurately link the position of the ablation to the specific season, such as a summer monsoon, when that portion of the ring was formed?
I mean, we are always guided when we are deciding on the resolution. We can either be agnostic and decide to use an equidistant or same resolution, but this is where the expert knowledge comes in terms of understanding tree growth informed by tree ring width and variability, and the climate and the ecosystem of the studied trees, where you can inform where to position that. So then you can link it, right? You can either use time markers, but then also we have the possibility in the laser ablation software to record the coordinates of each scan or ablation mark, and then we can, similar to quantitative wood anatomy, try to reposition that within one ring, and ideally link it to the position in the season. But that's, at least for now, what we can do with high precision is the early wood part and the late wood part for sure. And you can see from the images I showed you, and the nice thing is you can go back and revisit those tracks, and you know exactly where you are within the early wood and late wood portion.
Excellent. So greatly appreciated. I would like to thank everybody for taking the time to join us today, for the excellent questions and your attention. If anybody has additional questions, I'd like to encourage you to feel free to send them to us afterwards. My email address is lucas.smith@teledyne.com. You also have the email addresses of the other presenters. We have also recorded the presentation, so if anybody is interested in watching it over and over again, rest assured you'll be able to in the near future. Just let me know if you're interested. We'll also host it on our YouTube channel, and I'm sure that the CIRCON team will also post it. We do have a shared LA-IRMS website that we've been promoting as a portion of our collaboration, and I would encourage all of you to take a look at that for latest and greatest news on the technique, upcoming developments, that sort of thing. It's a really wonderful tool and also includes support information as well. So with that, thanks again to everybody, our presenters, and please look for details on future presentations.